Welcome to the Biosafety Level 3 facility at the Indian Institute of Science, Education and Research at Tirupati. This is a national facility created by ANRF, Anusandhan National Research Foundation. So this is one of our most high-end microscope that we have inside a BSL-3 in India. What it can do more than other microscopes that you find in other places is that it can take a live image for a longer time for a live bacteria or a live cells that is infected with bacteria or viruses, right? So you can see in real time how the infection is progressing or how the metabolic changes are happening within the bacteria and the viral infected cells, right? So we can also have an option of putting a microfluidic device here, and wherein we can do organoid cultures in future, right? So we can actually bring in miniature models of infections inside a microscope, right? So that's the specialty of this particular equipment that we have planned and housed inside this BSL-3. Yeah. This is our cell culture facility. Here we can actually culture cells which are infected with bacteria and viruses. So this part is for the bacteriology part section, and so we prepare the cells here, and once the cells are prepared, we infect them with the bacteria and then keep it here. If you like to infect them with virus, we transfer this to the next lab, which is a virology section, through this pass box, which opens only in one direction. I would not be able to open it in the other direction at the same time, right? So once the pass box is closed, there is an air exchange which takes place and sample gets into the other lab, right? So this is the infection biology lab of the BSL-3. I'll show you another most advanced equipment that we have housed in this facility, and that is a fluorescence assisted cell sorter, right? So this is the equipment which can actually sort, which means separate cells based on fluorescence, right? And one can have different fluorescent proteins being produced by these cells, which are at different stages of their life cycle, or you can call them metabolic state, right? So this is a very, very high-end equipment, and one can just put a sample in here, and then one can collect the cells in 96 well plate format, and one can actually separate cells in at least 4 different fluorescent colors that we would like to get. So that is something very unique which we have put inside a BSL-3. The other things that we require in a BSL-3 we already have, and I have shown you the bacteriology lab and the virology lab, where we would actually culture cells. These are for the in vitro biology labs, where we would use biosafety cabinets, and these are maintaining negative pressure in the room as well as inside. It's a unidirectional flow, so moment we take out a sample, we cannot open it outside. We have to take it inside, and only then we do all the manipulation that we need inside this in this airflow chamber, right? So we have 2 such units for doing the manipulation, and we grow the cultures after that in these kind of incubators, where these are shakers, comes incubator, and it shakes and allows bacterial growth, right? And we also have some static incubator there, and these are also environment control. One can maintain temperatures. We have a similar setup for the virology section here. I showed you the cell culture facility that will prepare the cells before infection, and now those cells will come out, the prepared cells will come out from this site, and this will then come out from here and then get inside this biosafety cabinet, and the viral particles will then be infected into the cells, and one can monitor the different stages of their infection as time progresses. So this is what we plan to do here. Thank you.